This is the last video in the immunology section of Unit 3 of the Higher Human course, and it's also the very last part of the whole of the course. It's really short and generally there's not really much that can be asked about apart from exactly what's on this. It's mostly related to problem solving, questions about vaccines or effectiveness of drug trials and vaccine trials. So mostly these questions will be to do with analysing data or a big experimental design question. Right, so the previous video that done covered vaccination and why it's important. This one covers vaccine trials and drug trials. And basically the whole point of this is to test the effectiveness and the safety of vaccines and drugs. So this could be any kind of pharmaceutical drug, cancer treatments, things like that, okay, or it could be any vaccine. So the COVID vaccine had to go through this trial or these type of trials, and all the drugs that you can get from a pharmacy will have to have done the same thing. Okay, so it tests effectiveness and it tests safety. Now there's 3 specific ways that these trials, these tests are designed to be, okay? First of all, they're designed to be randomized. Now you need to be able to explain why they are randomized, what's the purpose of that randomization, right? And explain what that means as well. Okay, so the subjects in that trial, okay, so the people in that trial are divided into random groups. Now you could describe an example to explain this if you wanted. So for example, they could have names drawn out of a hat. Okay, so there's no deliberate placement of any group of people or any types of people into a group. And this is important because it reduces bias. Right, so basically so that you don't get 2 groups, one with males and one with females. Now to kind of give you a more emotional example, if you think about cancer treatments and cancer trials, if you look at a young person and you think that's, as a specific example, a young person with leukemia, if you're looking at the trial, the effectiveness of a drug that treats leukemia, and you know someone that's young that's got leukemia, you want to make sure that they have that vaccine or that drug, okay? But that's not how these trials are designed. You can't place people into these groups because therefore you don't know if it's been effective. You can't allow your emotions to then determine who goes in which group. Okay, otherwise you might have more males or more females in a group, more older people in one group, more younger people in the other, or more likely, if you maybe know that someone's health is poorer, you might try to push them into the drug trial so that they maybe will have some benefit. Or if you know their health is poorer, you might not include them in that drug group because it might not be effective anyway. So you don't want to bias that in the way that you distribute the people in the groups. The next way that you have to design these is so that they are what's called double blind. Right? Now, double blind basically means that neither the subject, okay, so the people in the trial, or the researchers, right, the doctors that are running it, for example, know which group they're in. Okay, so know which group the subjects are in. So if a person knows which group they're in, whether they're getting the drug or not, that might influence how the results come out, okay, and how you would then interpret your results. If a researcher knows that a person is getting a vaccine or a drug, they might interpret their results differently. Okay, so it prevents biased interpretation of results. Right, that's really important that it's not just the patients, the subjects that don't know. It's really important that the researchers don't know. You can have unconscious bias, that you're not aware that you are looking at that in a different way, but it happens anyway. Okay, so it's really important that you have someone else out with that has had those groups randomly organised so that you as a researcher or you as a subject have no clue which group you're in. And the last one, which you'll probably have heard of in terms of the placebo effect, is what's called the placebo control or controlled groups. Now, basically that means that one group gets the drug or the vaccine, right? And one group gets a placebo. Now, a placebo is basically something that replaces the drug but will cause no effect. So, for example, a sugar pill rather than a pill that would be a drug. Okay? Or, for example, if someone's undergoing a chemotherapy, like cancer trial, then they would have an IV that would still go into their vein, but it would more likely be like a saline solution or things like that. Now, this is really important, I should probably write placebo control, because it allows you to actually validly compare your results. If you just had someone receiving a drug and then someone not receiving a drug at all, or not receiving anything, not taking anything at all, they know they're not getting the drug. Okay, but you will get placebo effects. Some people in the placebo groups will generally feel better, even though they've not received anything that's clinically effective. Okay, so this is really important because it allows valid comparisons. Okay, think about the term control. We know about controls from scientific experiments. This is to allow you to compare and to show the effectiveness of that drug compared to something that replaced the drug that didn't cause an effect. Right? And lastly, what's really important as well is that these groups have to be of a suitable size. Now, you can't just have 10 folk in a group. That's not enough, okay? Because therefore, if one of them has a bad reaction, that's 10% of the group having a bad reaction. But if you had that larger scale group and you had tens of thousands of people in it, and one person had a bad reaction, that percentage is tiny. So it reduces the size of experimental error as well. And it also makes the results that you have more reliable because you've got more and more people in that group. Now, if a drug trial has all of these aspects in it, okay, the drugs are then, the results from that drug trial or that vaccine trial are compared. Okay, and they're compared to determine if any differences between the placebo group and the drug group are what's called statistically significant. Now, that's basically just to show you that if you've got one group taking a particular pharmaceutical drug and one group taking a placebo, they both might get better. Okay, so you have to make sure that that is significantly increased in the kind of drug group more than the placebo group. Okay, otherwise we could just give everyone sugar pills and they could be fine. All right, so it's really important that you have all these aspects. Okay, and you should be able to define what each of these aspects means without trying to use the term within it, okay? So placebo controlled as much as you can. Obviously you can say placebo, but try and describe that you've got one group with the drug and one group without it, but with something else to replace it. Right, and that finishes off the whole of the higher human biology course.